populations, identification of polymicrobial isolates, sufficient number of viable cells in the sample study to be positive, and lack of antibiotic resistance data. But this, the last one, it's repaired day by day because we have it's available a new software for MDR isolates for diagnosis and epidemiological studies in healthcare associated infections. So maybe we have a future in this also. And this is the molecule that is in our laboratory. This is the Vitek, the other one used in the microbiology laboratory. Of course, we have the rapid screening immunochromatographic tests. The rapid tests, for shorten, the detection of antigens in the pathological samples. There are many types of pathological samples that in which we can detect some antigens. For example, the one that is very well known is the pneumococcus and Legionella pneumophila and Newt, but we have also in other type of diseases. The immunochromatography lateral flow assay is based on this antibody reaction. The advantages are shorter time, minutes, 10 to 15 minutes, high sensitivity and specificity. It's very simple. It needs a small volume and a minimal preparation, so no need for an additional device. It's a very rapid test made on paper. They were called in the past field tests, so you can do it everywhere. You need minimally trained personnel, long shelf life, monitor refrigeration based on very good. Of course, it has some disadvantages. Nothing is perfect. The qualitative or semi-quantitative results and needs confirmation. It's how no matter how positive is a test, this kind you have to confirm it with the other traditional methods. But it's very useful for us to start a targeted treatment. These are the, all about the rapid tests in COVID, for example. And the indications are not only for human medicine, we can use it also for veterinary medicine, environmental control, forensics, criminology, for screening, diagnosis, prognosis, monitoring. So we can use the immunochromatography, lateral flow assay, in many types of other domains. These are examples of etiological agents targeted in this kind of tests: malaria, Ebola, and fungal infections, and so on. We can use it in food domain. This is now not our domain, but it's you all know about the aflatoxin testing in food, for example. And another part of the diagnosis is serological diagnosis. Even if it doesn't mean the identification of a germ, it's easy and very available to apply. And it's the first line of diagnosis in suspicion of infectious disease, but it has severe limitations. The diagnosis usually is retrospective because the antibodies takes around 10 days to appear after infection. And the interpretation of these results is very complex. And you have to take into account all the aspect of the patient and all what he did, what treatment he took, what kind of vaccination he underwent in the last time, and so on. Types of serological diagnosis. ELISA, everybody knows about ELISA, yeah, immunoassay, immunofluorescence, immunoblot. They all target different type of antigens and antibodies, and make us to undergo the diagnosis finally to the real one. And the other domain is the antibiotic resistance testing. It's a very actually very important part of the laboratory diagnosis. The purpose is, for example, the screening for MDR, XDR, PANDR portage. We are doing this screening in our hospital at a targeted group of patients, the patients that had contact with hospital or other kind of medical care units in the last 90 days, for example. With this, we are doing this for isolation measures. Of course, this is a measure taken in pre-operative situations, and also for antimicrobial stewardship program, it's very important. And types of techniques for the detection of this MDR, XDR. So the most important ones and most used ones are the chromogenic media for the detection of multi-drug resistant bacteria. And it has advantages, reduces the number of colonies that must be identified in a mixed culture, shorter time, cost reduction, better detection rate, faster diagnosis, but does not provide absolute specificity, right? It's useful. And what's the main problem is that it must be identified by other methods after you have the chromogenic media. And these are examples of chromogenic media for different types of germs. As you can see, for salmonella, for streptococcus, for vibrio, helicobacter, candida, and MRSA, different type of microbial can give you more details about this type of chromogenic media. And what is the microbiologist and the clinicians doing? It is the total automation of the microbiology laboratory. It's increasing the quality of microbiology diagnosis, it's reducing the time to obtain the diagnosis, it permits a good management of a very large number of samples simultaneously, and the reduction of highly qualified staff. Of course, this means efficiency and economy, and we have, not in our laboratory for this time, but we know about examples of total automation microbiology laboratory. For example, the three ones Bedek-Yestra, Kupan, and Biomedical. I will not go into detail explaining this. They are containing different types of identification techniques. For example, this one with Monditov and other type of diagnosis. It looks like that, so it's a mini laboratory, or like that. Biomedical concept FMLA. It's a total automation concept of the microbiology laboratory. Modular design, you can change this depending on your interest in the type of diagnosis. It can do automatic blood culture, gram staining, so it's a combination of classical and new techniques, biomedical techniques. And it looks like that. And the third one, also with modular design, processes various samples, performs smells and gram staining also in combination with new techniques, Monditov, Bitec, and other types. And it looks like this. So it's a very small laboratory, completely automatic, and very useful for the diagnosis. Conclusions: the microbiology laboratory is essential in the infectious disease specialty. So it's the vertebral column of the infectious disease hospital. Without this, we don't have any diagnosis. Molecular diagnostic methods are an important support for us, and we are trying to keep it updated. The automation of the bacteriology laboratory is the future, and the human component remains vital for correct interpretation of results. This way we will have only a list of germs, and that's all. So we need the clinician, we need the microbiology specialist. Thank you.
Any short questions, maybe?
Well, if you have very special questions, I will try to ask my colleagues to answer if it is a very detailed microbiology question.
Or we could pass the question for the round table.
For the round table, yes. We are interpreting later.
Thank you very much. It's an honor to be here, the co-chair of this session, and now Professor Kirk and Mr. Monicev to rapid detection of pathogens and resistance contribution for cytometry. Thank you.
Just need a minute. After Dr. Florescu introduced us to the state-of-the-art presentation in the available methods in the microbiology laboratory, now I will try to come with a researcher point of view and show you our efforts in using flow cytometry for the rapid detection of pathogens and antibiotic resistance directly in clinical samples. Of course, there is a long way from the bench to the bedside, but we are at the start and we are very open to collaborate. Until they face, I can tell you that the antibiotic resistance is a big, huge problem. And I think that the challenge of antimicrobial resistance will be even bigger after the pandemic, COVID-19 pandemic will pass, because we have used a lot of antiseptics, disinfectants, and antibiotics in this period of time. And unfortunately, Romania is among the countries, European countries, with the highest rates of antimicrobial resistance because of a lot of reasons, but probably because we are using a lot of antibiotics incorrectly. And also, we are among in the top of the European countries regarding the consumption of antibiotics in the veterinary sector. And a lot of antibiotics used for treating human infections are also used for treating animal infections. And these antibiotics and their metabolites are released in the environment, in the aquatic medium, and they are turning back in the clinics, and they are changing resistance genes. So the phenomenon is very difficult to control and is very, very diverse and depending on the geographical characteristics. So our colleagues told you about a lot of PCR-related methods for detecting resistance genes, but I must tell you that these techniques need a lot of customization because the genes, the resistance genes are very different in different regions. We are saying that this is the reason for which some years ago, European ECDC, European Center for Disease Control, was funded because in order to have a local, let's say, authority to respond to the local challenges. Our colleague from our research group, Luminița Gabriela-Măruțescu, she is the specialist in flow cytometry. Actually, she is the vice president of the Romanian Association of Cytometry. And I made this presentation because she is representing Romania in another meeting in Greece now. Sorry. I've already told you about this. The antimicrobial resistance, the dimension of the challenge is very important for the public health. This is one of the One Health problems, actually, requiring a global and holistic approach. I'll tell you about the current state of research regarding the application of flow cytometry for pathogens detection and antibiotic resistance detection. About the commercially available flow cytometry-based methods for detecting for antibiogram, let's say, on short, and I will finish with our studies regarding the application of flow cytometry for pathogen detection and antibiotic susceptibility testing dealt in clinical specimens. So, talk 10 with antimicrobial resistance, and if we look here, we can see that if no action is taken, the cause of death in 2015 will surpass the number of deaths by cancer. Because we are not talking about the increased number of infections and the mortality associated rates, but also about a lot of modern medicine progress, like major surgery, transplant, cancer, chemotherapy, which are no more possible if this resistance continues to be at such high rates as it is now. So antimicrobial resistance is either genetic and it is transmitted vertically by mutation, but it could be also transmitted among the members of the same population, so very rapidly, by horizontal gene transfer. And we are talking here about horizontal gene transfer that could occur within 1 hour. So, if we don't have rapid results of antibiotic susceptibility testing, we are getting the wrong antibiotic, and this antibiotic will act as a selective pressure for that bacterial population, and in 24 hours, the entire susceptible bacterial population will become resistant. So, we are thinking that we are putting the patient on the antibiotic treatment, but actually we are killing him. So it is very important to have very rapid results of antibiotic susceptibility. And we are not talking here about detecting genetic material because the genetic material could belong to dead bacteria. We are interested in detecting the susceptibility of viable bacteria colonizing a certain patient. Besides this genetic resistance, we must also take into account the phenotypic resistance. So, here it is an image probably you know from a Star Trek movie. It is the Borg. This Borg population is adapting all the time by learning. The bacteria are doing the same they are learning, their experience with each antibiotic. And we can trace this experience and these memories in their sequences. We can see that by genetic sequencing, by whole genome sequencing, but it does not help us for treatment. It does help us for epidemiological surveys. For treatment, for correct treatment, we need rapid results of antibiotic susceptibility testing. And coming back to collective stress response, our bacterial cells are communicate each other, and they are trying to run from the antibiotic. They are moving away by motility, but also they are isolating themselves in biofilms, and these biofilms are protected by a matrix which reduces the penetration by antibiotics. And moreover, in this collective, the stress-induced mutagenesis occurs, and this leads to the occurrence of hypermutator phenotypes, and they are adapting very easily to the presence of an antibiotic, leading to accelerated evolution and adaptation. I was telling about the problem of resistance in Romania. You see that for many antibiotics and multi-drug resistance rates, we are somewhere between 25 and over 75%. And this is due to many problems, but we can at least respond to a small part of this big problem by the development of rapid and or direct, because it is not possible to have direct methods all the time, and accurate antimicrobial antibiotic susceptibility testings. Our colleague discussed about the currently available testing, phenotypic and genotypic testing, all of them with advantages and disadvantages, but excepting genotypic testing, the phenotypic testing takes time because we need to isolate the bacteria, we need to identify it, and then we make the antibiotic susceptibility testing. So the rapid antibiotic susceptibility testing will improve the therapy outcome because many times, particularly in the hospital, the clinical condition of the patient is very severe. So we are giving antibiotics before having the results of antibiotic susceptibility testing. And I was telling you about this very rapid adaptation in a matter of hours. So we need some rapid antibiotic susceptibility testing that could give us results in a very short time, so a few hours after the admission of the patient in the hospital. If it's possible to have these results together with hematology and biochemistry results, the treatment would be more adapted to the clinical context offered by the patient. And there are a lot of novel methods for the rapid phenotypic measurement of growth. So rapid phenotypic measurement of growth, because we are interested to see the response, the real-time response of bacterial population to a certain antibiotic. And I will refer to flow cytometry. Why did we choose flow cytometry? Because in the lab, in the clinical lab in the hospital and ambulatory also, the clinicians have access to flow cytometer because they are using flow cytometry hematology. Okay, so we were discussing with the clinicians, and they said that they are open to use the flow cytometer to make this rapid antibiotic susceptibility testing. The principle of a flow cytometer, I know there are specialists in the room, so the synthesis is that by using combined fluidic, optical and electronic instrumental approach, we can get information about the size of the cells, about the metabolic activity of the cells, about the granularity of the cells, and also we could label using fluorescent dyes different components of the cell and get also more information and many parameters regarding the behavior of a single cell or of a population to a certain, in the presence of a certain antibiotic. And flow cytometry started to be applied in microbiology not very long ago. First in biotechnological processes to control the metabolic activity of starter cultures, for example, in beverage industries. After that in environmental microbiology, and last in clinical microbiology. And the principle would be sampling, and we are intending to standardize the methods to detect directly in clinical samples, and not after we have isolated the strains, because this isolation requires at least 10 hours. And then the pre-treatment, because we are talking about very complex biological matrices here that could influence the results. And then staining, this is optional because now the researchers are trying to standardize label-free methods in order to not interfere with and to not bring artifacts in their determination, and then the multi-parameter analysis. And as fluorescent dyes, we can use also for labeling the microbes the same fluorescent dyes that are used for marking the mammalian cells, like fluorescent dyes for membrane potential, for efflux pump activities, for enzymatic activity, or for membrane and bacterial cell envelopes permeable integrity, because this is a difference between membrane potential and membrane integrity. This membrane integrity, alteration of membrane integrity suggests, for example, bactericidal activity of an antibiotic, while membrane potential modification or changing would suggest only a bacteriostatic activity, and this is important also for the clinician. In the literature, there are label-free cytometry methods for detecting antibiotic susceptibility phenotypes in pure cultures. But pure cultures means that we have already lost 24 hours. Then also other type of tests using also label-free cytometry. So when we are saying label-free cytometry, many of you from this room know very well, means that we are getting information about the size of the cell by interpreting the side scattered light, and also about the granularity of the cell by interpreting the forward scattered light. And these parameters could be sufficient in order to differentiate between viable cells, metabolically active bacterial cells, and dead cells. Because dead cells are changing their size, are changing their dimensions, and other things like this. The artificial intelligence and machine learning could represent a very important progress in the future, because the results from the flow cytometer would be interpreted and according to databases, existing databases, we can make a classification of the bacterial cells in susceptible or resistant, depending on this aspect on the flow cytometry dot plot or histogram. What I've shown you until now was referring to bench site research. Now we are getting to commercially available flow cytometry based rapid tests for pathogens detection and susceptibility testing. And there is a bacterioscan, commercially available test, allowing the direct detection of uropathogens. So in urine, it is not by happening that this was the first test that was introduced, because urine, and our colleagues from the microbiology laboratories could confirm, probably it is the most frequent clinical sample analyzed in the laboratories. The uropathogens are detected based on their size, refractive index, shape, and density. Another commercially available kit is the Fastinov. This Fastinov is adapted for blood culture. Positive blood cultures means a very severe clinical landscape, clinical infection, and needs immediate treatment and correct treatment. So here you can see in 2 hours, Fastinov would give the results regarding the antibiotic susceptibility, while the classical approach is much longer, 2 to 3 days. Now I will pass to our results. So these results were obtained in the frame of a research project. It was an IDEAS research project, rapid flow cytometry assays for microbial detection and antibiotic susceptibility directly in clinical specimens. We have started this project because our lab is involved together with our colleagues from different hospitals in the country in the epidemiological surveillance of antimicrobial resistance in Romania. We have a lot of PhD students working on clinical sector, but also on the environmental sector, trying to fill the gap, let's say, regarding the circulating resistant bacteria in our geographical territory. Also, the possible transfer between clinical and environmental reservoir of resistance. And starting with these strains, these collaborations, we have an important collection of resistant strains, over 3,000 bacterial strains, which are very well characterized by phenotypic and genotypic approaches. Hundreds of them are already whole sequenced, so we know a lot about their resistance genes. And this was the starting point to move a little step further and try to bring a small contribution to the standardization of a rapid method to detect these resistant bacteria in clinical samples, being convinced that this would mean something for the improvement of the empirical anti-infectious therapy. We are calling this emergency antibiotic therapy empirical therapy, because it is not based on the results offered by the lab. We are not saying that these rapid methods would replace completely the standard methods. No. But we have this first step of treatment, and after that, after receiving the results from the laboratory, following the conventional diagnosis, we can change the treatment. But at least know that we don't kill the patient, giving him an antibiotic that is acting as a selective pressure for resistance. Okay, we were starting with urine samples, so we have chosen 27 uropathogenic E. coli strains. We have first characterized the strains by conventional methods, and then analyzed them for antibiotic susceptibility testing using our Accuracy 6 class flow cytometer. We have it in the lab for microbiology. We have other flow cytometers in the lab, but this is very friendly, let's say. It's very easy to move. Also, we can put it in the laminar flow cabinet, so it has a lot of advantages. And we are using we have chosen ceftriaxone, ciprofloxacin, nitrofurantoin, and trimethoprim sulfamethoxazole. These antibiotics are used frequently, and the resistance rates to these antibiotics in the strains circulating in our country is quite high. So we have chosen different concentrations for these antibiotics, which are characterizing resistant and susceptible strains, and we put the bacteria in contact with these concentrations of antibiotics and then analyze them in flow cytometry. So in 4 hours, this was the analysis time. We here used urine samples, but not, there are many steps. I won't present here all the steps, but it requires a lot of time and commitment because we have started with an artificial formula of urine, then with urine from healthy people, and then with real urine samples, and this is quite difficult because, as I told you, the biological matrix is very, very complex and it's very different from patient to patient, and this influences our results. And you can see here we have used a quite high size of the bacterial inoculum. We have chosen it as a threshold because the microbiologists know very well this is the threshold for differentiating between positive urine culture and cultures and negative urine cultures. Let's say what we have obtained. Let's see what we have obtained here. So, first we have gated the viable population. This is quite challenging because the dimension, the size of bacteria is very low, a few micrometers. So for example, for someone who is acquainted with flow cytometer and is using flow cytometry for analyzing the blood cells, this is the green. They don't take it into consideration. So we gated the viable E. coli population depending on the site scattered and the fluorescence of D-bag C. So this D-bag C fluorochrome is a marker of membrane potential. After that, we followed this fluorescence and calculated the median, the staining index. So the staining index is the ratio between the median fluorescence of treated versus untreated control. We are talking here about antibiotic-treated bacteria. And the ratio must be higher than 2 in order to consider that the respective bacteria is susceptible to a certain antibiotic. And we could represent the results like this. So in few hours, the gray field histograms, the untreated control having certain fluorescence, and then with the blue line, the bacteria population incubated with different antibiotics. And all these antibiotics at different tested concentrations are active on the bacterial cells. So they are affecting the membrane potential. So the fluorescent dye enters in the cell and the fluorescence is shifted to the right side. So this is the aspect of a population which is susceptible to all tested antibiotics. So we can tell the clinicians in 4 hours they are bringing the sample to the lab, and in 4 hours we are telling him which of the antibiotics from the emergency panel, because not all antibiotics are used in this emergency panel, are active on the respective bacteria. But we could also have this aspect. So, as you can see, the fluorescence of the treated bacteria is practically superposed over the fluorescence peak of the area of the untreated bacteria. So, the antibiotic does not affect the bacterial population. And the results would be susceptible to nitrofurantoin and ketomoxazole and resistant to cyclophosphamide and ceftriaxone. Very, very rapid result. Or if we are doing that for the epidemiological surveillance, we can represent our results like this. And here, with this horizontal line, we have that threshold of staining index. And what the strains which are under the staining index are resistant to the respective antibiotics. The strains which are over the staining index are susceptible to the respective antibiotics. And we can examine every week or every month these histograms in our hospital, and we can see the evolution of antibiotic resistance in our hospital unit or in our lab. And we can then communicate with the pharmacist, with the infectionist, and maybe leave apart some antibiotics for which an increasing antibiotic resistance is observed in time. After that, the biggest step and the most difficult is to validate our results. And these results are validated using the conventional methods. Respectively, we have used gross microdilution, so we have used a quantitative method to validate our results, and we have obtained some false negative and false positive results, but when putting the numbers in the formulas for sensitivity and specificity, we can see that overall the sensitivity and specificity of our protocol are quite good. Taking into account that we bring these advantage of very quick guiding of the clinician, we consider that we can move forward in order to see this protocol in the clinics. So here, after we have tested our protocol for detecting the bacteria in the urine samples and testing their susceptibility, we have focused on certain phenotypic resistance phenotypes. So maybe some of you have heard about ESBL, extended spectrum beta-lactamases. These are very powerful enzymes which are rendering the bacteria resistant to almost all beta-lactam antibiotics. And the beta-lactam antibiotics are very important class of antibiotics because they are very effective, but also they are not toxic because they are acting of a certain target of bacterial cells, the cellular wall, which is missing from the human cells. These strains are resistant to antibiotics, but these enzymes are inhibited by the so-called ESBL inhibitors. And such an ESBL inhibitor is the clavulanic acid. So we have tested these ESBL producing bacteria in the presence of ceftazidime, so ceftazidime is not active, and in the presence of ceftazidime, the third generation cephalosporin combined with the inhibitor of ESBLs. And we are expecting that the clavulanic acid is inhibiting the enzyme, and the third generation cephalosporin will become active. And as you can see here, indeed, the violet line is the line, the fluorescence line corresponding to the bacterial cells incubated with the antibiotic and with the inhibitor of the resistance mechanism, and we can see how the fluorescence is shifting to the right. So the bacteria is recovering its susceptibility to these important antibiotics. Then we have passed to blood samples. I'm telling now very easily we have passed, but this stage was the subject of a PhD thesis. And another PhD student of mine had these blood samples because in blood samples is very important to have a quick diagnosis, and also sample preparation, sample staining. Also using, this time we have used two fluorescent dyes, one for membrane potential and one for permeability. So propidium iodide and 1-oxanol. And the steps were more or less the same, but it was quite a challenge because the composition of the blood was very rich, and we have centrifuged the blood, and we have worked on the plasma because bacteria are very small, and they are at easy centrifugation. They are staying in the supernatant. So we have taken the supernatant, added the fluorochromes, and studied the aspect of the bacterial population. Here we can see the results for resistant isolate, one E. coli strain resistant to ciprofloxacin. Gating, so we have marked our population of interest based on size, scatter, and fluorescence, and then we have followed the intensity of fluorescence on different channels. Here we have the channel for protidium iodide, and here we have the channels for DPAC. So combining this information, we have decided, we have found that this strain is isolated, is resistant to ciprofloxacin, because you can see these very small percentages of fluorescence, so the dye does not easily enter the cell, meaning that the cell is resistant. This E. coli strain was resistant to ciprofloxacin by hyperactivation of an efflux pump. We know that because I told you that we have very well characterized the strains. And here, ciprofloxacin is susceptible E. coli isolate, and you can see the percentages, the much higher percentages of fluorescence, particularly for DBAC. But the total number of viable cells, you can see, is very, very low, because the majority of cells are dead. So here in this gate, you see that the percent of the viable population is very low. And here different aspects of the histograms of the dot plot, obtained in cerebrospinal fluid samples. So we have chosen this type of clinical specimens requiring immediate and correct antibiotic treatment. And we can see the aspect of the histogram to different antibiotics, to different concentrations of antibiotics. And I think that now you can easily say which of these strains are susceptible or resistant to different concentrations of antibiotics. So we can guide the clinicians not only in telling them that the bacteria are susceptible or resistant to a different antibiotic, but also guide the clinician to choose the active concentration to be given to the patient. Here, another form of representation of our results for many strains, and as I told already, the depolarized bacteria have a low staining index and are represented by these small columns. And our last PhD student is involved in testing the usefulness of flow cytometry for detecting susceptibility to last resort antibiotics. We are calling these antibiotics like fosfomycin, tigecycline, ceftazidimab, ceftolozane, tazobactam, and colistin. We are calling them last resort antibiotics because it doesn't mean that they are new. Some of them, like fosfomycin, are revived antibiotics. They were excluded for many years from the treatment because of their side effects, but they have been introduced again because we don't have any choice. MDRO, meaning multi-drug resistant organisms. And another example of such a revived antibiotic is colistin. And you can see some plates, some antibiogram plates from our hospitals is not very encouraging, what we can see here, for example, or here. And the protocols for the detection of resistance to these last resort antibiotics like colistin are very
endemic, mainly infectious diseases emerged and one pandemic, one large pandemic, which mess with our life for 2 years now. I will start the story about electron microscopy. At the beginning of 20th century, Victor Babeș came in Romania and developed an institute of bacteriology and pathology. He was one of the most important pathologists and bacteriologists at that time, and wrote the first textbook of bacteriology along with his friend Cornel. And he was interested in almost all infectious diseases, with specific interest in rabies, which was quite frequent at that time. He described some pathological lesions specific for that disease, and also applied therapy, anti-rabies therapy. Vaccine was settled by Pasteur 2 years later, and Babeș already started to use. Even the pathogen was not known at that time because the techniques was not available. The virus was supposed to be the pathogen because it was too small to be filtered. Was also non-replicative in culture media, and required cell culture to replicate. So we need to thanks to Ruška brothers for electron microscopy. In fact, Ernst Ruška designed the first electron microscope in 1931, and his brother was interested in viruses and bacteria and asked for improvement in this first model, and Ernst did that. We have here the first paper showing you some viruses. Mousepox, in fact, is here. Looks like a nanoparticle you are working with. But meantime, the electron microscopy developed, and the technique and the software and the new added technologies of ice producing in a vitreous state improved a lot the electron microscopy. Why to use electron microscopy to identify an unknown pathogen? Because it's quite easy, quite fast. 10 minutes will be enough to see a viral particle using negative staining, a specific technique. You can see 2 or more different infectious agents at the same time in the sample, if this is the case. Does not require a living organism. In fact, processing the samples for electron microscopy requires a fixation, and good aldehyde is killing almost everything. We know we need some reagents, of course, to increase the contrast in electron microscopy images, but not something special like antibodies or PCR products, nucleic acid standards, or and so on. We have no false positive results, and this is a good thing. And we can see, in fact, the infectious agent, and we can classify the pathogen. We have some limitation in electron microscopy. We need a quite large quantity of pathogen in the sample, but usually when it's an infectious disease with unknown pathogen, the lesion will be quite dramatic, and a lot of particle will be available in the biological sample. If it's not present, this is not equal with the absence of the pathogen. And we have some limitation linked with the expensive instrument, which is the electron microscope. And extensive knowledge which is not always available. We need a biologist or microbiologist trained to recognize the pathogens in this kind of techniques. Transmission electron microscopy is the best way to look for viruses mostly, and bacteria of course. And we have available some techniques from classical plastic embedding procedure, which is quite laborious and time consuming if we need to look for infectious agents in tissues or cells, to the quite easy to do negative staining technique where the particles will be bright on the dark background and easy to recognize. And new techniques which involve the specific equipment and a specific technique to obtain the vitreous eyes and to embed the particles inside. But the resolution is quite good, allowing us to see molecular details at atomic level. Negative staining is a technique which is quite easy and fast. In 10 minutes we can have the grid prepared for the microscope, and 20 minutes to examine the grid will allow you to put the diagnosis. This is the only support which could be introduced in an electron microscope, a transmission electron microscope. So we can put a little drop of biological fluid containing the particles. We can let the grid to dry, and after that we cover the particles which are on the grid with a small thin layer of metallic solution having large atoms. This solution will prefer to stay in lower places surrounding the particle and creating a dark background for the particle, which is quite shiny in this technique. Mostly phosphotoxic acid gets used for negative staining, and this technique was used for identifying almost all viruses in 60s. June Almeida is one of the researchers who was interested in viral classification, and he wrote a poem for this technique, negative staining. This is her image took in 1966, having a coronavirus unknown, zoonotic origin. But also she tried to classify the viruses according with their location into the cell, into the nucleus or into the cytoplasm, according with their size or their morphology. And this classification is still in use today. Even we have nice cartoons emphasizing the structure, the morphology, the different morphology of different types of viruses. Most of them are quite nasty for humans. All this information comes from electron microscopy. Negative staining or classical transmission electron microscopy, we have all these morphological details like size, shape, internal structure from electron microscopy. So the electron microscopy is quite a good technique to discover the new infectious agents. In the last 10 years, maybe, the electron microscopy changed a little bit. We have a new type of electron microscopy, cryo-electron microscopy. The liquid nitrogen temperature is the temperature which is quite stable inside the microscope, and we can see viral particles or any kind of molecules frozen in a thin layer of vitreous ice in a state most appropriate from native states. So we have no need to put different heavy metals or chemicals to have a contrast, because we have this technique which is sustained with quite sophisticated softwares which allow us to have a nice image of particles, molecules. In this case, influenza virus is quite visible, trapped in the vital signs. This kind of technique is used today to detail at the molecular level, at the atomic level, all the molecules composing the viral particles, and we have some databases like this one, Protein Data Bank, which is holding the informations acquired through cryo-electron microscopy, and electron microscopy data bank, also a repository for data obtained from cryo-electron microscopy and cryo-electron tomography, in fact, because we can do tomography. We have 3 types of microscopes in Victor Babeș Institute. The institute built for Victor Babeș, in fact, when he came in Romania. We have a small one they use for diagnosis, for mostly for kidney biopsies diagnosis, and 2 other more complicated, and one with cryo facility, cryo capability. Our experience is not quite large with viruses or infectious diseases, mostly with kidney diseases. We work with kidney diseases, and sometimes we have kidney transplant biopsies, and transplant biopsies involve some immunosuppression, and these patients have an immune system compromised, and sometimes infectious agents could be developing in their kidney. Even quite benign polyomavirus, which can build some large nuclear inclusion, like this one here. If we enlarge the image, we can see the particular structure, and even closer, we can imagine, mostly in this transmission electron microscopy, the echohedral symmetry of the virus. In the transmission electron microscopy sections, when we do the sections from plastic embedded tissue, the section is about 100 nanometer thick. So we have the entire particle in the section, and even more, and sometimes we have different layers superimposed in the same 2-dimensional image. But in this kind of technique, we can see viral infection, and also associated with bacterial infection. And sometimes this diagnosis could be unresolved in clinical laboratory. Another experience we have with viruses is the negative staining of different inactivated influenza viruses for vaccine producing protocols. So we can do negative staining and measure the particles which are changed by different experimental conditions, and the morphology, the changed morphology could give us some important data. This experience was before we had this electron microscope with cryo facility, and we resumed the collaboration with our colleagues from Cantacuzino Institute for vaccine producing with cryo-electron microscopy. In some conditions, it was supposed to have a change in a particular matrix protein in influenza virus, and this modification of M1 protein, it was suspected to generate a quite fragile membrane for influenza virus vaccine. And we tried to see in cryo-electron microscopy if the changing solution for inactivation viral particles will change the structure of the membrane. So we measure the membrane of the influenza viruses in different conditions, and this is in cryo-electron microscopy. One big problem for that is that influenza virus is quite polymorph. We have small particles here and larger particles in the same solution, in the same sample, and the membrane could be different in bidimensional images. So we decided to do electron tomography, cryo-electron tomography. This involved to tilt the grid inside the microscope and acquire serial images, reconstruct the volume. And this is the tomography, one of the tomographies, and you can see entire particle in the volume reconstructed. From this reconstructed volume, we can extract some digital sections which are very, very thin and could measure the membrane. From tens of cryo-electron tomograms, we extract some particles and made some libraries containing thousands of particles, and measure the membrane in different conditions, in different experimental conditions. And for, sadly, we have no good results. The two conditions we had investigated didn't change the membrane, the structure of the spike protein of the or the M protein, matrix protein, and this happened in 2020, and everything changed because at that time a new pandemic outbreak with coronavirus 2 emerged, and our colleagues in Cantacuzino Institute were diverted in different other tasks. This is the first image of coronavirus 2, SARS-CoV-2. We have clear spikes around the central particle, and this image directed the research to the coronavirus to be a culprit for the pandemic 2020. After this image, of course, all the molecular techniques Professor Flores already presented you started to be used to generate a test, a specific test, sequencing the genomic material, give us the PCR test, and so on. So some coronaviruses were culprit for endemic outbreaks of infectious diseases and this pandemic in 2020. We have here a negative staining images of various coronaviruses, SARS-CoV-2, SARS-CoV-1 in 2003, and a transmission electron microscopy showing you particles inside a cell. In transmission electron microscopy for plastic sections, the corona glycoproteins will not be visible because of due to the processing of the cells for transmission electron microscopy. Cryo-electron microscopy was useful studying SARS-CoV-2 virus because detailing the atomic structure of the spike protein was important to predict the variant shifting or to decide if new antibody designed specific for specific three-dimensional structure of the spike protein will be effective in the therapy. We had a vaccine quite quickly for this infection disease for SARS-CoV-2, and we did a negative stain for the two types of vaccines available today. The classic one with TAN adenovirus, very nice particles visible in this image, and for the new type with synthetic RNA in a lipidic shell. They are looking very nice, very clean, very beautiful, and for sure electron microscopy would answer this question without no problem. No, the COVID vaccine cannot connect to the internet to anybody. We made a small experiment giving a vaccine in cell culture to a certain type of cells, and we observed the specific, the classic route of internalizing the vaccine through endocytosis and endolysosomal compartment specific was filled with this ribonucleic particles. We observed after 12 hours that some very few cells exposed on the surface. This image here is a magnification from this one. Few cells exposed on the surface, some spike proteins, which could be in a living body exposed to the immune system and trained to generate antibodies. So this kind of extension, cellular extension, is quite strange and not usually seen in cell culture, but you can see here very nice spike proteins in cell after 12 hours. This is a team of my lab, electron microscopy lab. Emmanuel is the one who performed the testing of cells in culture with vaccine administration, and he tried to reconstruct the spike proteins from the cryo-electron microscopy images. We worked in collaboration with Professor Onwu and his team for influenza vaccine, and we have a lot of students from medical faculty to working with us for image processing, especially because it's time consuming and require quite a good knowledge of the software, so they are interested in this kind of things. So this is the story I want to tell you about the electron microscopy as a useful tool for diagnosis of unknown infectious agents, mainly viruses, but also I believe will be a good tool for the study of bacteria and their viruses, fungi, because the microbiota it's a new topic in medical field, and we know very few things about this type of cell in our body. Thank you.
Thank you very much. We have time for a question. people.
First thereafter.
Okay. And now, our last invited speaker. We are honored to have here Otto Larsson with inverse problems and hierarchical multi-scale modeling of biological matter.
Yeah,
Welcome.
that's correct. Which one? Oh, this one. Okay. Okay, thank you. First of all, I would like to thank the organizers and Professor Simeon Esco for inviting me here. It's a great pleasure to be here. I'm going to tell you a story which is quite different from what you have heard. Today it's about in silico studies, and this title is basically also the outline of my talk. So I will first tell you something about inverse problems and how to solve them. Then I will focus on hierarchies in biomolecular structures. And then I will introduce a method we developed in our group, which is quite general, but I show examples on how to use it to study biological matter. I should of course say I come from JAS Intercentro. I'm in the group of Dr. Maria Pikala, and I just want to say that I really enjoy to be here. There is an incredible atmosphere, scientific atmosphere, interdisciplinary, and I have learned a lot in working there. Otherwise, I come from Stockholm University. I did graduate there, and I also did all my scientific career, except all the breaks I did abroad. And also now, once in a while, I go to Nanjing Tech University. I'm a member of staff there with some teaching and supervision, and also I'm a visiting professor in the most northern university in Sweden, Luleå. It's very close to polar circle, and this picture is taken in the middle of December, where there is basically no daylight, but you can see very often beautiful northern lights there instead. Okay, so about inverse problems, actually you all, what I've been hearing today, when you do imaging, you solve inverse problems, you don't reflect so much about it. But otherwise, if we stop thinking about it, so when we solve inverse problems, we invert what we know, find something we don't know, and then this way a solution is the inverse of the problem. So normally we solve forward problems. So here is a very simple equation. Actually, it doesn't need to be the linear line crossing the origin. X can be some very multifunctional equation of the data structure, and K is an operator. So finding the inverse, solving the inverse problem, then we have to find some kind of inverse of this operator. But to solve inverse problems uniquely, is typically very difficult. And this depends on inconsistencies and noise in the available data, and also the parameter space is often very, very large. You have to explore. But we solve continuously inverse problem. Every day we are trying to understand what just happened and why. And of course we use common sense and all life experience to try to figure out that. In science, we very often use two things which are not so easy to define: educated guess or reverse engineering. So this lady is just studying a test tube after a synthesis, trying to find out what did I get here actually, and then you can do reverse engineering, and modeling is often very useful in reverse engineering. Okay, in nature, all living species, birds, snakes, whatever, they are solving inverse problems to find food and also to survive. They use, they rely on perceptions, like sounds and light and smells and so on. In science and mathematics, solving real inverse problems is common. There are scientific journals for this purpose. And currently it has become a kind of hot topic because it's boosted very much by machine learning and big data. I'm sure you've been watching this TV series, Crimes in Investigation, and it's about solving forensic inverse problems because they have to reverse the time to this point of time when the crime did happen, and sometimes you get really impressed when you watch this TV series, how they use all kinds of science from biology to whatever, and they immediately make very fast conclusions. And so, then, yeah, solving inverse problems can also save your life. No. Okay, so about hierarchy. Nature prefers hierarchy in structures it develops. Nature is the most superior material scientist there is. And one good example is the genome, starting from DNA and how it is packed in nucleus and core particles and chromatin and so on. And this first structures are very well known today and studied, and also the chromosome, but then there's a gray area here which we still don't know so much about because we have to take into account the dynamics also, and also at different points of the cell cycle, different things happen there. So I will come back to this a little bit later in presenting you some multi-scale modeling, which is the topic of my talk. Nature also prefers composites together with hierarchy because it's this way you can nature can create very strong structures easily, and then these structures can be expanded and they can grow and so on. This is a piece of our skin which also can be seen as a as a hierarchical structure, and it can be modeled as well. So all these structures are really ideal for hierarchical multi-scale modeling. So now I should say something about multi-scale modeling. But first, modeling is also called in silico studies. It's computations, it's studies taking place in a silicon chip, and so on. And when you do in silico studies on biological structures and processes, there are very many challenges there. And one is, of course, the incredible number of orders of magnitude in length and time scales and other scales. And then finally, it's the biology itself, which is built successively in the matter. And it requires very specific conditions, so it's very complex, diverse, and there's so much chemistry and energetics and thermodynamics, etc., etc. And finally, in silico studies, it's completely beyond reach life itself, self-organization, and so on. But we can try, at least on this point, to do modeling. So, if you look at the so-called nanoscale, it starts perhaps from 1 angstrom and it goes to 100 micrometers. It turns out that when you do in silico, there is no single modeling method you can use to cover all this nanoscale. Typically, you can start with so-called first principles methods, quantum mechanics, quantum chemistry, where you create atoms and molecules from nuclei and electrons. Beyond that, you go to classical mechanics. You use something called molecular mechanics, which doesn't contain any more electrons, but you have atoms, and from atoms you build molecules and molecular systems, liquids and solids, and so on. So you wonder where these limits come from. They are actually only practical limits given by the computers and the computing power we have at the moment. So it means that the limits are moving slowly to the right when computers are becoming faster and faster. After that, we have soft matter and mesoscale, and we can forget about the individual atoms and molecules, and we treat aggregates of them. And we can study soft matter. We can also study organelles, and chromosome was one of the examples, and different types of complex biological soft matter. So here are a few examples of these structures. I will come back to them later. So now I introduce this method which we are using, which we developed actually a long time ago. And it is based on something called successive coarse graining. And in every coarse grain step, we solve this so-called inverse problem. So the entire method is called the method of inverse Monte Carlo. It actually combines very well with something called multi-scale imaging, because you can do imaging also at different length scales and so on. So they combine very well together. So let's start from these first principles simulations. I will not explain these methods, so this means Born-Oppenheimer molecular dynamics, this means Carr-Parrinello molecular dynamics, this means tight binding molecular dynamics. They're not they're not important here when I describe this this method, but they are methods to perform simulations to treat molecules and how molecules move and their dynamics. And as we saw in the previous slide, we cannot use these first principles methods for very large systems, so we have to start with limited size of systems. So we do a simulation, and now from results from these simulations, we solve the inverse problem. We create a model which we can use on the next level. But when we use this model, we get exactly back the same results we got in this original simulation. Doing this, we throw away all the degrees of freedom we don't need in this next level. And going from quantum mechanics to classical mechanics, we don't have much use for the electrons, so we throw them away. Now, here we can increase the size of the system like 2 orders of magnitude because we don't need to treat with electron degrees of freedom. And we do exactly the same. From results we get from here, we can create a model we can use again on the next level, which is the mesoscale. And here we throw away lots of things which we don't need here. we throw away the atoms, we throw away the bonds, we throw away, for example, solvent entirely. So we just have particles there. And this way we can go up several levels. We can co-scale the co-scale thing, create a new model, go to the next level, create a new model, and so on. And this is the level many of the methods experimental methods we heard today also come from top down. So this is obviously from bottom up approach. And then what we can do is we can also go back. This is called fine graining. We have to try to reinvent these degrees of freedom we did throw away. It's not trivial at all, but it can be done to certain systems. If we want to go further up, we have to abandon the particle description of the matter, and then we can use elastic networks, finite elements, lattice Boltzmann, and other types of methods which are not based on particles anymore. But this methodology here, it can be used, for example, in non-equilibrium simulations, like for red blood cells flowing in the capillaries. It can be used for drug transport and delivery. Cellular uptake can be modeled with these models and many more applications. So this experimental techniques also, we saw many of these videos today with particles moving slowly around. They can be used to create these soft matter particles, and also the interactions can be estimated from that. And then this can be used in simulations further. Okay, this inverse Monte-Carlo method, it provides something called solvent-mediated interaction pair potentials. That's a force field. That's something everybody who has done molecular simulation knows what force field is. So possibly not all of you know, but it's like an interaction law, how particles, when they come close to each other, how they feel each other, and what type of forces they use to against each other. And so we use structural information. We can take it either from experiments or also from very accurate theoretical simulations. And the secret is that there is a correspondence between something called radial distribution functions, GOR, and these pair potentials, this force field. And historically, we can go back early 60s when already the first time these GORs were inverted and potential mean forces of them were calculated, potentials for liquid metals. The real breakthrough came in the mid-70s when Henderson did prove a theory that there actually is a uniqueness between radial distribution functions and pair potentials. This means that when we solve this inverse problem, we can solve it uniquely. He also showed later that it is true for multi-component systems. However, this theorem doesn't say how to obtain one from the other, so this has to be invented. So our inverse Monte-Carlo is based on something called Svensson's Monte-Carlo renormalization method on Ising models from late 70s. There are also other similar structure-based methods, and I would like to mention one from Florian Müller-Blatte. It's called iterative Boltzmann inversion. In fact, we use it always initially because it's a very simple method, and then we go at some point we go to our own method to get better convergence. Okay, so it's based on structure, this, and I like very much water molecules. They are among the simplest molecules there are, but they are also the most complicated molecules. And here is a group of water molecules. You probably can see it, and they're very happy. They decide let's take a selfie or a group it, and then one water molecule then keeps this stick with the camera, and this camera has its shutter open all the time. That means that the images are overlapping, they come on top of each other. And when you look at this bunch of water molecules, it also applies for all liquids. Liquids are very chaotic structurally, but there is still a structure there which we don't immediately see. But now suddenly we can see the structure in this groupy photograph. And it is because when this one particular water molecule keeps this camera, this stick, he also creates a molecular coordinate system on itself. So it means that this water molecule stands still, whereas all the other molecules are moving. And this picture also shows that this water molecule donates 2 hydrogens to these other water molecules, hydrogen bonds, and also it accepts 2. So this could be already the structural information we use. But we do something else. We use these radial distribution functions, which actually is the same thing, but not instead of in the molecular coordinate system. Now we calculate the distances and interactions in a laboratory coordinate system. So we get the function like this. There's a first maximum, there's a second maximum, and this is the function we invert to this pair potential. Radial distribution gives the probability to find another atom close by at a certain distance, and then it is normalized to bulk density. And this function can be obtained from molecular simulations or also from diffraction studies, electron or neutron diffraction. But you will get it as a structure factor in a K-space, which you have to Fourier transform to real space. And then first. Okay, so this, I think I will skip it. It describes a little bit the theory about this inversion, but what we use is Newton-Raphson. We start from the reference, the accurate radial distribution function from simulation, for example. We calculate potential of mean force, and we use it as a potential to calculate the radial distribution functions. And in the first iteration, yeah, I would say the black curve here is the reference, the exact. But the first iteration, we get something which is completely off. But you see, after 10 iterations, it converts to the original radial distribution function. And when this happens, we get a coarse-grained potential function which we can use. And this we can use at all these levels in this ladder you saw previously, and we can calculate the potentials. So I give you some examples here. Here is water in water. So you see here is the OO distances, but there is also OH and HH. And when you invert this, you get pair potentials. These are actually not coarse-grained potentials because it is exactly like a new water model. But if you study like sodium chloride, table salt in water, you have the contact peak for sodium and chloride, you have the solvent separated and so on. And when you invert this, you get potentials which look like that. They don't look like Lennard-Jones or any simple potentials. They are quite ugly and they oscillate. But that is because they contain the hydration shells. So we can use these potentials without water to do the simulation, and in a few seconds we get these radial distribution functions which took several weeks when we had all atom potentials there. Okay, so we can go to more complicated systems like phospholipids. We invert the selected radial distribution functions, we get effective potentials, which we can use in coarse-grained models, and so on. If also you notice that at long distances, these potentials, even they oscillate first, but then they coincide with Coulomb's law. So this makes that you can divide these potentials to short-range parts and a long-range part. And of course, the short-range part is very ugly, wiggling functions, whereas the Coulomb's law is very smooth. If you use this method to simulate, for example, DNA, you have to decide the topology and how do you co-screen and so on. So this is an all atom description of DNA, and this is a very simple co-screen model. This is where there is one nucleotide per bead. Here we have sugar, phosphate, and base as beads, but then we have to decide topology. Then you can use it in simulations like this, where we have studied up to 500 base pairs, twist and drift of DNA, mini circles, and so on, which you probably have seen in all-person telephone. However, this is which shows how useful this method can be. So, if we start from a system where we have all the atoms there, including water molecules and the counter ions around DNA, when we co-screen this, in the next step, you see there are no water molecules or ions anymore, because now they are in this effective co-screen potential. We can then use this to create nucleus and core particles. Here actually the interior is made of elastic network model. And then you successively co-screen it and make the system bigger and bigger. And this is one piece of this chromatin, and it contains only 98 beads. And if you go back and describe this on all atom level, you would need 11 million of atoms to describe exactly the same piece. And this way we come quite long in describing the genome. What is left here is this gray area again, which if we want to go further, we should use a very very simple, let's say, polymer models or something, but then we should combine it together with experimental methods like fish and Pisces and so on, and then we can go further. So this is something we are working on at the moment. So this should be a very good example of systematic hierarchical multi-scale modeling from DNA to chromatin based on solving immense problems. So this is my last slide also. So thank you very much.
Very much. It was really invigorating. Yes. So try to the attention possible. So there are some questions.
Yes.
Please.
Thank you. It was a very nice presentation. I enjoyed it. I'll try to read your paper. I didn't respond. I didn't know about it, so I should. The problem I would like to ask you, so the method of getting from the pair correlation function back to the potential is working only if I have spherical symmetry for the pair correlation, no? So you, no, sorry, for the potential. So usually the two-particle potential, what you are looking for, it has also direction dependence, and then the inverse problem should not work. Yeah, going from the pair correlation function back to the potential, no?
Yes. Well. So what we do, we create the asymmetry what you're referring to.
Yeah, there is direction dependence on the potential.
We don't actually have it because we have, like in atomistic simulations, we have pair potentials. So the pair potentials are between two particles, and using enough different pair potentials, you also describe this asymmetry or whatever. But within these pair potentials, there is uniqueness between between pair correlates and
Okay, so
functions.
if you consider only two particle interactions and not the three,
Yeah.
then it is working.
Yeah, but of course there are other problems which I didn't mention, and because it is based on structural information, so you should not expect that you get dynamical properties correct, and because it gives two soft potentials, it also gives two fast dynamics. But if you use these potentials in Langevin dynamics, then you can choose the friction coefficient. So you calibrate it with experiments
And also the noise, you can take it out, the noise properties from it. Because in the Langevin equation you have also the noise, not only the friction.
Yeah, sure. But of course it's not supposed to be that accurate as all other simulations. But when you start, you can actually start from first principle simulation, and that means that no empirical information goes in. But of course, we start from all atom level using some force field, and then this way, when we go step by step, we take with us the atomistic information. So for example, this Martini force field model with your planetons potentials, it doesn't have these details as we have in our model.
So why do you call it Monte Carlo? Because in your scheme was nothing random, is there? It was a totally deterministic.
Actually, in a paper we call it reverse Monte Carlo, and then we noticed a couple of months later that there was already a reverse Monte Carlo from Hungarian colleague. And then we started to call it inverse Monte Carlo. Then now we call it actually Newtonian inverse. So I should have used that. Okay. More questions?
Other questions? Thank you for your wonderful presentation.
Unfortunately, we have just 20 minutes, 25 minutes for the round table. The bus will come in about 20 minutes. So indeed, right. Most likely we'll start now, but we'll have the round table at the
Would you like to comment put some subjects on the table? Or?
Well, I think I'm the last one to put any subject on the table. Okay. What I could do is to ask some questions.
Okay.
For instance, I was excited by all the presentations, and I may put some questions from a naive perspective. For instance, because one of the challenges that are faced by medical doctors, not by the physicists, for instance, what is how to manage persistence. So these very rare type of cells which manage to escape antibiotics. Do, for instance, PCR molecular methods, are they able to reveal the presence of such capability of such this type of bacteria, for instance, or flow cytometry? Is it possible? Are they able? Do they have the sensitivity for pinpointing these type of cells. Another type of question, so most likely I am just asking questions, most likely we'll discuss the dessert or something like that related to this. I was also trying to understand why most of the results related to the amount of the bacteria we have, if there is an amount which is below 10 to 4th bacteria per mil, then it is stated as negative. And then I understood, or I thought that this could be related to the capability of normal people, let's say, of their immune system to stand such amount of bacteria. And if this is the case, then it has to be a relationship between the susceptibility to a specific antibiotic and the capability of the immune system of an infected person to respond to infection. And if this is correctly put in place, then it has to be another type of analysis revealing exactly this, so the capability of the immune system to react. Is there such an analysis usually performed? Questions? If not, how to relate this, let's say, diminished capability of the immune system to another threshold, 10 to whatever, 3, I don't know. So, as a physicist, I can ask questions.